Hi, welcome back to Educator.com. This is the lesson on metabolism and nutrition. So with the basics of metabolism, the word metabolism is often misused by people I've talked to. Usually people just talk about metabolism in terms of, oh, she's so lucky, she gets to eat whatever she wants, her metabolism must be really high. There is truth to that statement, but people only think of metabolism as keeping weight off, or metabolism the other way, gaining weight. But metabolism, the actual definition of it is that it is the sum of all chemical reactions within an organism. So it includes all the breakdown of nutrients that you need to do through digestion and all of the building of molecules that you need to do to maintain your body and stay healthy. So metabolism involves a basic formula that would be catabolism plus anabolism. So the catabolism part is the breakdown of organic molecules. So every time you break down a sugar that you've eaten, a lipid that you've eaten, which is a fat, a protein that you've eaten, that's a catabolic kind of reaction, and it would involve the release of energy. Anabolism is the opposite. It's the synthesis of new organic molecules, which typically requires energy and energy input to get those things linked together. So when you build muscle proteins, when you make glycogen, which is a sugar storage molecule that's mostly found in the liver, those are anabolic reactions. So the anabolism part, what are you doing it for? Well, it's for structural maintenance and repairs, for support of growth. So as you grow, of course, you need to build more molecules. For secretions, you need to build hormones, you need to build secretory products that you're going to be letting go through glands. And then for nutrient reserves, like I just mentioned earlier, glycogen. If it's been hours since you've eaten, you can release sugars from that giant storage molecule that we call glycogen, and that gets you through until the next meal. The way that I remember the difference between catabolism and anabolism is I remember the term anabolic steroids. So if you follow sports especially, you hear about anabolic steroids that athletes will use to bulk up their musculature a lot more than they could naturally without it. And the reason why it's an anabolic steroid is it is a molecule that mimics testosterone, and it allows them to have a lot more anabolic development of their muscle tissue. They are building a lot more muscle proteins than is natural. So anabolism, anabolic steroids, the building up of organic molecules. So we look at nutrients that we take in our diet. There are four major organic compounds in terms of looking at them and their structural differences and what they're for. Carbohydrates is one of them. They're also called sugars. Lipids, also known as fats. Proteins, one of the more important ones, and they're all important, but proteins make up the majority of your solid matter. If you took all the water out of an organism, the next most abundant substance is going to be protein. Extremely important for cellular structure and functionality. And the nucleic acids. You can't deny the importance of nucleic acids. That's DNA and RNA. And it's interesting to have that under nutrients, but anytime you eat animal tissue, plant tissue, even if it's dead, you're going to have these molecules in there that you are also going to consume. And you can break down those nucleic acids to make your own nucleic acids in terms of stringing together new DNA, new RNA. Vitamins and minerals. Vitamins, definitely also an organic compound. The reason why these are separate is vitamins are those molecules that you don't have the ability to make enough of them on your own. You do have to take them into your diet. For instance, if you stop taking in vitamin A early on in your life, that could cause your eyesight to suffer. But people who have enough vitamin A in their diet, they may gradually need glasses, but in terms of their eye health, it's going to be maximized by having enough vitamin A. So you do have reserves of vitamins typically in your body, but if you don't take them in your diet, you're going to run out. And then minerals. We'll cover these towards the end. Minerals you get from electrolytes, stuff like sodium, potassium, etc. Oh, and before we move on, this picture right here, if you're wondering what that is, these are a kind of packing peanut. Like if you got a big box with something shipped to you, instead of using styrofoam peanuts, these are actually made of starch. So this is entirely natural. You could eat them if you wanted to. It's kind of like those puffy cheese snacks without any flavoring. But yeah, completely natural, a biodegradable replacement for styrofoam packing peanuts. Carbohydrate structure. So we just talked about packing peanuts made of carbohydrates on the previous slide. Carbohydrates, these are sugars. And the interesting thing about the name carbohydrate is that when you look at the carbon atoms in one of these molecules, it's surrounded by water, H2O, H and OH. So together, this is water. Together, this is water. Carbo, like carbon, hydrate, like water. So it's an easy way to remember the structure of a carbohydrate. And this is the typical basic sugar structure in terms of a little ratio. N is some variable, so we could put 6 in there. Okay, so for every 6 carbon atoms, you're going to have twice as many hydrogens and the same amount of oxygens as carbons. We could put an 8 in here. We could put 100 in here. Any integer will work. So you can have slight variation of this depending on the precise sugar you're talking about, but typically, more often than not, you're going to see this ratio. So if you counted these up, you would see that. If there were 6 carbons, it could be glucose. If there are twice as many, you're going to get something like a disaccharide, which is like 2 little glucoses or monosaccharides put together. You're going to see the ring or linear form. This is the ring form down here, of course. It looks like a little ring. This would be known as a hexose sugar because it has 6 sides. Glucose is a hexose sugar. This is the linear form. You tend to see the ring form more often in the human body. It's more stable molecularly than the linear form, but you can occasionally see this. And the way you go from linear to ring or vice versa is carbon number 1 would connect to carbon number 6, and the double bond here would actually go away when you make that connection. Carbon makes 4 bonds. If you took chemistry, you know that carbon likes to make 4 bonds. That's why with every carbon here, you're going to see 4 little lines around it, whether it's a double bond or single bond. Over here, another way of writing this is the H could be sticking out here, a little hydrogen sticking out here, and then the OH or hydroxide here. But this is a shorthand version. Speaking of shorthand, when you look at this ring form, this is classic organic chemistry. Every corner here that's not labeled as O is carbon. So there's a carbon here, carbon, 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 carbon. And what they're leaving out is the H. It's just implied that on the other side of this OH is going to be an H. On the other side of this OH is going to be an H. Same with over here. So if you're wondering why is there stuff missing, that's why. So monosaccharides are single sugars. Glucose is the classic one. Disaccharides is a double sugar. So we could connect these 2 glucoses to make something called maltose. Maltose is a disaccharide. That's where you get malt sugar from. And the way it happens is you make a glycosidic linkage, which is just a fancy term for saying a bond between 2 monosaccharides. And the way you do that is through a dehydration synthesis or dehydration reaction. And the way that I remember that term is all you need to do is take water out. That's why it's dehydration. So here's an H, here's an OH on the next sugar. When these 2 leave together, H2O is gone. So that's why it's dehydration. And what you're left with is this oxygen. So if I then erase what has left, effectively, the water is gone. The way that you get this glycosidic linkage is through that oxygen connection. So you're going to have carbon connected to oxygen and then connected over there. Now the angle, because of this drawing, is not precise, but that's what you're going to see as a glycosidic linkage, is you're going to have this oxygen atom linking up the 2 neighboring sugars. And so a disaccharide includes maltose. Fructose is another one, which you're going to find in fruits. There's a lot of other disaccharide names, but those are a couple. Polysaccharides is when you have bigger sugars. You keep putting little sugars strung together, and you're going to eventually get things like starch. So examples of polysaccharides, starch, which is the way that plants store sugars. You also have glycogen. Glycogen is how we store sugars in our liver and muscles. Starch and glycogen, both made of lots and lots and lots of glucoses. It's just the way that the glucoses are bonded together is slightly different in terms of the way the glycosidic linkage looks. And then another good example of a polysaccharide is chitin. I know it looks like chitin, but chitin, you find that in the exoskeletons of arthropods and also in the cell walls of fungi. You also can use chitin to make surgical threads that you can sew up something surgically, and they actually will dissolve. Those chitin threads will decompose eventually, which is kind of cool. So dehydration synthesis is how you get two sugars together. The opposite, like if I wanted to break these two sugars apart, is called a hydrolysis, and that's made of hydro and lysis. Hydro means water, lysis means a breaking apart or splitting apart. So water splitting them apart. And it's the opposite of that dehydration reaction we just went over, that if water left to connect these two, water going in can also do the opposite. It can separate them. Sugars are water soluble. That's obvious. If you put pieces of bread in water, you see them dissolve pretty easily. Pasta, when we boil pasta or some other noodle, it's very obvious that sugars, which make up the majority of pasta, it's water soluble. It is a polar kind of thing rather than non-polar, which is the lipid side. And hey, the purpose of carbohydrates in general is they are a really quick, efficient energy source. That's how we get the most of our energy molecules in our cells. ATP is kind of like an energy currency that fuels our cells and keeps us alive. And the easiest way to get that is breaking down carbohydrates or sugars. So we need to talk about aerobic respiration. This is how you actually get energy out of the breakdown of organic molecules and keeping your cells going. So it's the process of breaking down carbohydrates or other organic compounds that you can sort of turn into a carbohydrate-like molecule with the help of oxygen. That's what's aerobic about it, that you need to breathe in oxygen to make ATP, adenosine triphosphate, which you're going to hear more about on the next slide, and that is the energy molecule. There are other molecules that are similar, but ATP, very common. Most of the process occurs in mitochondria. So if you took biology, you learned that mitochondria are kind of the powerhouses of the cell. That's where you get ATP from typically if you're eukaryotic like we are. So here's a mitochondrion. This is an actual electron microscope taking a picture of mitochondria in human cells. This is sort of your classic computer-generated model of what a mitochondrion looks like. And if you look carefully or closely at this particular diagram, you can see that the outside is a double membrane, inner and outer membrane. The inside, you have these membranous folds, and it's called cristae. And then on the inside, you have the matrix. So you do have this two sets of membranes here. That's why the space in between the cristae and the inner part of the double membrane on the outside is called the intermembrane space. But we're going to cover these parts because aerobic respiration, it starts out outside of the mitochondrion, but then it ends up going in. Speaking of the term mitochondria, that is plural. Mitochondrion, with an O-N at the end, is singular. So the three main parts of aerobic respiration, using oxygen to break down sugar to make ATP, is glycolysis, which is the initial breakdown of the sugar molecule, the Krebs cycle, which takes the products of glycolysis and goes through a cycle to help make other molecules that are going to transfer energy to the end of the process, and the end of the process is oxidative phosphorylation, where we're making a lot of ATP, the energy molecule. And by the end, you're producing approximately 36 ATP. The reason why it's approximate is depending on the conditions inside the cell at that moment. Sometimes it could be 34, sometimes it could be 38, but generally we get 36 ATP from one glucose molecule being broken down with oxygen. So what is this ATP molecule? So ATP is adenosine triphosphate, ATP. And here's a model of it. So where does the name come from? Well, this part here, you got these two rings of carbon with nitrogen and some hydrogen thrown in there. You have adenine. And adenine is a nitrogenous base that you would find in DNA and RNA, but here it has nothing to do with genetics. You're using adenine as kind of like a placeholder for what's going on here. And then, what is this? Well, this is ribose. a kind of monosaccharide, simple sugar. It is a pentose sugar because it's 5-sided. But adenine, ribose, and then 3 phosphates. So here's where the name comes from. Adenosine. So the combination of these two, adenosine. Instead of saying adenine, ribose, 3 phosphates. Adenosine triphosphate, 3 phosphates. So that's where the name comes from. And you're going to see the word or abbreviation ADP also on the slide. That's when one of these phosphates is gone, and then it's diphosphate. So adenosine triphosphate is kind of like a charged battery that you can use to power something in a cell. When you go back to ADP, adenosine diphosphate, the battery has no more juice in a sense. The other analogy I've heard is like a rubber band. If I stretch the rubber band, that's ATP. If I let it go, it goes back to ADP. And to stretch it back again, you got to add that 3rd phosphate. You do see AMP in cells as well, adenosine monophosphate, but we're going to talk a lot about how you get ATP from ADP in the next few slides. So like I said, it's the energy currency of a cell. You can spend it in terms of powering anything that needs to happen in a cell that requires energy. So ATP is broken down into that adenosine diphosphate plus the other phosphate. So when ATP breaks apart, you get these two, and that's a release of energy. That's how you spend the currency. To get it back, you got to put these back together, and that requires energy to attach them back together. And that's how you get ATP. So up here, when you break it down, that is an exergonic reaction. Here, it would be endergonic, the input of energy. And amazingly, the average muscle cell, which is doing a lot of work in a typical day, doing a lot of movement, of course, the average muscle cell produces 10 million ATP molecules per second if it's working. That's amazing to think about. That's mind-boggling that a microscopic cell is producing millions and millions of these molecules every second just to keep it going. All right, so to start aerobic respiration is glycolysis. And the word glycolysis, glyco, lysis, glyco meaning sugar, lysis meaning breakdown. Like the word lysosome. It's one of those organelles in a cell that break stuff down like foreign bodies, pathogens, waste. In this case, we're breaking apart a sugar. Glucose is usually the one. And pyruvate is what you get at the end here. That is a 3-carbon half of glucose in a sense. And it occurs in the cytoplasm. I've read that the average glucose molecule doesn't easily fit into mitochondrion. It's broken down into these two, which then enter the mitochondrion to finish the aerobic respiration process. So let me take you through this step by step and do my best to simplify it. So phase 1, what you're looking at here is these lavender, magenta, whatever, purple dots, whatever you want to call them. These represent the carbons. So we're focusing on the carbons of glucose. Glucose contains 6 carbons. What you're not seeing here is the H's and O's, and that's fine because we're concentrating on the carbon count here. So to initially get it started, they call it the energy investment phase. And you have to invest 2 ATPs. Now remember, ATP, that's usable energy. The phosphates come off of it to give you 2 ADP. And then you can see in the next shot, these little yellow dots, they're inorganic phosphates. That's why it's PI, not pi like 3.14. This is an inorganic phosphate, and they came from these 2 ATP molecules. Then you get a splitting of this glucose in half. So now G3P, I believe, stands for glyceraldehyde 3-phosphate. You don't need to know that, but so you now have kind of like a half a sugar with a phosphate attached to it, half a sugar with a phosphate attached to it. And the next thing that happens is some of the hydrogens are stripped off of this, and you end up getting what's called NADH. So initially, you have something called NAD+. I'll explain this. NAD+ is like an electron carrier. Think of it as a boat or shuttle for electrons, and when it takes them somewhere and the electrons go into a reaction, the flow of the electrons can power the making of ATP eventually at the end. So this is kind of investing to get way more ATP way at the end. So NAD+, when electrons, or hydrogen, which contains electrons, comes onto it, you get NADH. So this is like the powered NAD+ that's going to dump its electrons later on. And so you make this in the next few parts of aerobic respiration for the payoff at the end. You're going to see at the end, the NADH will give up the H, protons and electrons, to help make a lot of ATP at the end. The next thing that happens is you now have a vacancy here. Remember, carbon always likes to have 4 bonds. And something is left, the bond is gone, and there's free phosphates around. They're put into place here. So now you have these little halves of glucose, each with 2 phosphates. And the great thing is, in the next step is where you get some payoff. Remember, this was an energy investment. You had to invest 2, but you know, at the end, you get 4 ATP, and it comes from attaching these 4 phosphates onto 4 ADPs. And then you get 4 ATP. And the great thing is, 4 ATP, more than what we invested initially. 4 ATP minus the 2, still a net gain, total gain of 2 ATP. So just breaking down sugar gives you a little energy in itself, but we're going to get a lot more later on. In addition to the making of a net gain of 2 ATP, the 2 NADHs, you also have 2 pyruvates, also called pyruvic acid. So these 2 pyruvates each undergo a slight modification and enter what's called the Krebs cycle. So 2 Krebs cycles happen, 1 cycle for each of these, to continue the process of aerobic respiration. And these pyruvates, they're going to be going in the mitochondria. So the Krebs cycle, also known as the citric acid cycle, because the first part of the cycle involves the making of citric acid, but a scientist named Krebs, this is the one who named it after himself, and it's in honor of him. And the Krebs cycle, you can sometimes see it labeled as TCA cycle, the citric acid cycle, but usually these are interchangeable. So pyruvate gets modified into what's called acetyl-CoA, and CoA stands for coenzyme A. It's shuttling this acetyl to the Krebs cycle, which is in the mitochondrial matrix. If you remember from the previous image, here's a mitochondrion, if we looked at a cross-section of it. The Krebs cycles, they are happening in here, the mitochondrial matrix, which is within that cristae. The glycolysis stuff happened outside here, happened in the cytoplasm. And those pyruvates come in, and there's a lot of action going on here. Krebs cycles galore. So it takes 2 pyruvates, attaches them to a 4-carbon molecule called oxaloacetic acid or oxaloacetate, and then eventually breaks that down, the citric acid down, to make more electron carriers. And you actually do get a little ATP out of it. So what initially happens, let's use red for the carbons. So here's pyruvate. And eventually, you're going to get this out of it. So what happens is pyruvate is modified to make this little acetyl. And what happens is you get, let's use a blue box. This is coenzyme A. This helps modify and take pyruvate into the Krebs cycle. Once it's here, by the way, the little blue box leaves and goes and picks up another pyruvate. The other little pieces that happen here is you do make some NADH. So in addition to a CO2 leaving, that's how you get from pyruvate with 3 carbons to this little acetyl, which was attached to here, to acetyl-CoA. That's how you get the 1 less carbon, is CO2 leaves. And guess what happens to that CO2? We exhale it. you're going to see a couple CO2s leave in the Krebs cycle as well. And that's waste that you're going to inevitably breathe out. That's waste from the process of breaking down sugar. So here's basically what happens. This is how it gets modified to give you the acetyl, which is helped to get modified from the CoA. And this joins in with a 4-carbon molecule to give you, remember that's called oxaloacetate or oxaloacetic acid. This is citrate or citric acid. Yeah, the same citric acid you'd find in citrus fruits, but we are producing this constantly, every second of every day of our life in Krebs cycles. The cool thing about remembering the sequence here is that the first 2 steps are identical in terms of what ends up happening. The first step is that, and by the way, these are all catalyzed or they are helped to occur from enzymes. So these are enzyme catalyzed reactions. They don't all just happen naturally. This breakdown doesn't just happen unassisted. There's enzymes, proteins that help all of these things happen in sequence. So the first step is that CO2 leaves. And guess what? That happens again in the second step. CO2 leaves, and we'll eventually get down to the 4-carbon molecule again. It's not quite the same as this because some other things will be stripped off of this molecule, but you can see that once we get to this point, no more CO2 is leaving because at the end here, we have to come back, regenerate this 4-carbon molecule. In the first 2 steps, also, you make some NADH. Yay! The more NADH you make, the more ATP you inevitably will produce at the end. So see those 2 steps in the beginning, basically the same. Next thing that happens, you make an ATP. I'm simplifying this a bit. So ADP comes over and gains a phosphate to become ATP. Yay! Where does the phosphate come from? Well, there's a molecule, GTP, that helps get the phosphate into place. So there are actually a few little mini steps here that I'm glossing over. But the end result from this part of the Krebs cycle is you get the production of 1 ATP. So each Krebs cycle does give you another ATP. That's great. Next step, we make something called FADH2. FADH2 is very similar to NADH. The hydrogens that FAD gains, it's going to dump off in the last part of aerobic respiration to get a lot of ATP. By the way, FAD, NAD+, what do those stand for? I don't remember. They are long names, and that's why we have abbreviations for them. So FAD, also a electron-proton carrier, or raft, if you want to think of it that way, and now it's carrying that stuff. It stripped them off of this molecule. One more step, and then we are home free. Next thing that happens is you get one more NADH, and we're going to add up the products, the stuff that we take away from this in a moment. We wouldn't count this as a product, even though we're left over with this 4-carbon molecule, oxaloacetate, because it needs to go back into this Krebs cycle. It's not something we are taking away from the cycle. What we are taking away is how many NADHs? 1, 2, 3. You could count this one as well. A lot of people refer to this as the intermediate step between glycolysis and Krebs cycle, but you do get a few NADHs. You get 1 ATP, 1 FADH2, 2 CO2s. This is a waste product, but it is a product nonetheless. So the Krebs cycle, important step in the process of aerobic respiration. And next, we're going to the cristae of the mitochondria to actually make a load of ATP. Oxidative phosphorylation. Oxidative, you may think, oh, because of oxygen. That's fine if you want to remember that, but the oxidative part, when you oxidize something, you're breaking it apart. Electrons are coming off of it. When you reduce something, electrons are gained. So oxidation, reduction, or redox reactions is another way to put it. You learn about those in chemistry. If you get those mixed up, I remember that since electrons are negatively charged, when you say that something is reduced, you're adding electrons, so more negatives like reduces it in terms of like if you think of a number line. Oxidative or oxidation, that has to do with the opposite. You actually are breaking something apart. So NADH and FADH2, they're oxidized in this process. They lose what they gained when they were reduced, and they end up helping to make a lot of ATP, and that's phosphorylation. When you are adding phosphates to something, you are phosphorylating it, and you're phosphorylating ADP to make a lot of ATP. So now all these NADHs and FADH2s that were made in glycolysis in the Krebs cycle, you actually use them here. So you can see that here's our NADH that was made in the mitochondrial matrix. This is a simplified version of the cristae membrane. It's just a rectangle here, but typically you'd see it kind of weavy. But anyways, all along the cristae, that inner membrane of the mitochondria, you're going to have these proteins embedded in the cristae, in that inner membrane, and they're going to be shuttling along electrons. And what happens to the protons is all these H pluses, that's a proton. H plus is just a proton. So NADH gave up protons, so does FADH2, and they go across the membrane into the intermembrane space, so you get a buildup of all kinds of protons here. It's like, what's going to end up happening is it's basically kind of like diffusion of protons in a sense that's going to be happening. And they actually nickname it chemiosmosis. Chemiosmosis is a fancy term for the flow of protons that's going to end up going through this ATP synthase to make a lot of ATP. And the electron transport chain is the other part that happens here. And I'm going to use blue to highlight that. So all along here you get electrons, and electrons, their final destination is actually going to be oxygen. They call oxygen gas here, and then finally we're hearing about oxygen in aerobic respiration. They call oxygen gas here the final electron acceptor. It also technically is accepting protons too, because you end up making water. It's like the opposite of photosynthesis. In photosynthesis, water is broken down and give you electrons, protons, and oxygen gas. Here it's the opposite. Oxygen gas is accepting these electrons moving through, and then you get water. The water that's produced here in aerobic respiration is not enough to hydrate a human being. I've heard that some animals in extremely arid, dry conditions can go for days and days and days without drinking water. And for them, the amount of water that they're producing in aerobic respiration is enough to keep them hydrated. That's not true for us. But water is a product of aerobic respiration. So what happens to the protons? The protons build up in here, and they are going to be fueling directly the making of ATP. It's like water flowing through a water wheel. The water turns the wheel, and then you end up getting something being powered. The flow of electrons through ATP synthase, it's kind of like a turbine that spins, and as it spins, it helps attach phosphates to ADP, and you get a load of ATP, close to 30 ATP from doing this. And that's amazing to consider, because we got a net gain of 2 from glycolysis. We got a total of 2 from the 2 Krebs cycles. So the vast majority of ATP you're making during oxidative phosphorylation. And then, yeah, once you have NAD plus left over and FAD, that's going to go back and into the citric acid cycle. Some of it will exit mitochondria. There's probably actually already enough NAD plus outside the mitochondria for glycolysis, but nonetheless, the point is that the cycles keep going. As long as you're supplying glucose, sugar, and oxygen, you're going to keep having aerobic respiration occurring. So what happens when there's not enough oxygen It's called anaerobic respiration. So after glycolysis, keep in mind, does not require oxygen. There was no oxygen involved there. After glycolysis, the initial breakdown of sugar, if enough O2 is not available, pyruvates, the product of glycolysis, undergo fermentation. And what we do as animals, or human beings specifically, is lactic acid fermentation. There's also alcohol fermentation, which we take advantage of when we make alcoholic beverages, or even when we bake bread. Yeast do alcohol or alcoholic fermentation. But we do lactic acid fermentation. So lactic acid is formed after glycolysis if there's no O2. And that's what causes a burning sensation in our muscles. If we go for a long run or do a lot of intense activity in a short amount of time and you end up being out of breath and you get that burning sensation in your muscles, it's lactic acid. The more active you are, like if you're an Olympic athlete, you're probably not feeling that lactic acid burn because the efficiency of your body has been improved over years. But lactic acid, research has shown that, it makes sense that it would kind of slow you down. It's like, my muscles hurt. But what lactic acid also does in your bloodstream is it actually stimulates you to breathe more, to take more breaths, which is going to get more O2 in so that inevitably you'll be able to stop doing that anaerobic respiration and go back to aerobic once you've gotten a little rest. So fermentation, specifically the lactic acid variety, produces only the ATP from glycolysis. No additional ATP happened from fermentation. That means that anaerobic respiration as a whole, the total amount of ATP is just a net gain of 2. There's no Krebs cycle. There's no oxidative phosphorylation because, there's no making of ATP during this fermentation process. So here's lactic acid fermentation, glucose initially. Glycolysis happens, you get the 2 pyruvates or pyruvic acids, and here's the real fermentation part. NAD is regenerated. It's the opposite of what happened in glycolysis. We made NADH. So you can see that the NADH formed from glycolysis gives back the H's it gained to make NAD plus again. And then once you add that stuff from NADH back to pyruvate, you end up getting 2 lactic acids, or here they call it lactate. And that's what causes that burning sensation in your muscles. You can do anaerobic respiration for a period of time in your muscles, no problem. Anaerobic respiration in your brain, that's a problem. Your brain, without enough oxygen gas for several minutes, can result in brain death. So something like your muscles can deal with anaerobic respiration. Other tissues, their demand for oxygen and need for oxygen is even more. All right, so I mentioned earlier that you could have other organic molecules be broken apart for energy besides sugar. Now, sugar is the classic one that is broken down, but you can end up making glucose through gluconeogenesis from other organic precursors. Like, you could take lactic acid and make glucose out of it. You're probably going to need 2 lactic acids because lactic acid has 3 carbons. Glycerol, which is a part of lipids, it actually has a similar beginning to the name, glycolysis, glucose, glycerol. Glycerol is also 3 carbons. And you can take amino acids. The way that you take amino acids, the building blocks of proteins, and make glucose out of it is deamination. You're taking off what's called an amino group, which makes it an amino acid. So deamination is one example of how gluconeogenesis can happen. So you're taking an amino acid here, and through the help of an enzyme and adding water, you end up getting NH3. This right here is an amino group on an amino acid taken off, and then this resembles a sugar much more, and it can easily enter somewhere in that aerobic respiration process. Sometimes you're modifying it to make glucose. You can also sometimes modify a molecule so it can become an acetyl and go straight into the Krebs cycle. But either way, gluconeogenesis is a way that you can end up getting energy through aerobic respiration from other organic molecules. Glycogenesis would be making glycogen, a huge polysaccharide, a storage kind of sugar, from itty-bitty glucoses, the monosaccharides. And then the opposite, glycogenolysis, remember lysis is the breakdown. This is the breakdown of glycogen to make glucose. So here we're making the storage molecule, here we're breaking apart the storage molecule. Insulin is a hormone that gets sugars out of your bloodstream and packaged away as glycogen. So glycogenesis would happen thanks to insulin. Glycogenolysis would happen thanks to something called glucagon, which you can hear more about in the endocrine system lessons. So now moving on to lipids, lipid structure and function. They're called fats, and a classic example is triglycerides. So you can have a fat that's not a triglyceride, like cholesterol. Okay, that's also a lipid, that's also a fatty substance. But triglycerides are the classic one that you hear a lot about in textbooks. So what is a triglyceride made of? It's made of a glycerol attached to 3 fatty acids. That's where the name triglyceride comes from. So glycer, from that word glycerol, 3 fatty acid chains attached to it. So the glycerol, similar to a sugar, here is the glycerol portion right here. You see the 3 carbons, hydrogens attached to it. And initially they have little OHs here. That's what is the term, all like alcohol. They have OHs, hydroxides attached to them. But what happens to the H? Well, when you attach the 3 fatty acids on here, that reaction makes it so that actually it's just oxygen attached to a carbon, oxygen attached to a carbon. Remember from before with those ring structures, the ring shape of the glucose. At the corners here, the shorthand version is, hey, these are carbons. What's attached to the carbons? Hydrogens. So the other name for these fatty acid chains, which I'm going to use red for, is hydrocarbon chains. So here's a fatty acid chain. Here's a fatty acid chain. They can vary in length. Depending on the precise fatty acid chain that's attached to it, because they all have different names, you get different triglycerides. There's a lot of different triglycerides. Lipids are non-polar, opposite of water. They are not soluble in water. Just drizzling oil, like vegetable oil, in a pan of water, it's easy to see that. They don't like mixing together. So this is the only organic compound as a whole that's always non-polar. And what are these for? Energy source, insulation, and hormone synthesis. You actually can get a heck of a lot of energy out of a lipid molecule. Per gram, you can get more ATP out of it than sugars, actually. Insulation, think about all the lipids under your skin at the lowest parts or deepest parts of your integumentary system. It's a cushioning, it's also insulatory in terms of helping to keep you warm. And then hormone synthesis. There are some hormones without fat in your body you would not be able to make. Estrogen is an example. That's why women who end up having a very, very low, like dangerously low body fat percentage, sometimes will, their menstrual cycle will stop completely because they've stopped making enough estrogen to keep it going. And over time, that's not very healthy. Oh, and one more thing before I move on. Right here, you see an additional line. This is a double bond. The double bonds here make little kinks in the fatty acid chains. And that's going to come up in a little bit with the difference between saturated fats versus unsaturated fats. Here we go, saturated versus unsaturated fats. So saturated fats have no double bonds in the hydrocarbon chains. So when you don't have double bonds in that chain of hydrocarbons, so just carbon, 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 with H's attached all over it. If you have no double bonds, you don't get little kinks. The double bond can make it kind of bend. So they're all flat. When they're flat, I like to think of it as like this, okay? This textbook, all of these pages, these hundreds of pages, are all very flat with respect to each other. They're not bent, they're not folded, and so it's easy to close the book and make it very dense. Think of a lot of saturated fats as being that way. So saturated fats are typically solid at room temperature, like butter. So why is butter solid? It's because of the saturated fat that you end up getting from typically animal tissue. And if you have a lot of those saturated fats stacked closely together, they will be denser, they will be more solid. Unlike unsaturated fats, which have at least one double bond in the hydrocarbon chains, and the double bonds make it kink. And here's the example I'd like to use for that. Here's a flat piece of paper, which, like I said earlier, if it was saturated fats, they could be stacked really close together. But imagine I make it bendy like this. Okay, now these are representing those double bonds causing kinks in the chains. If I lay a bunch of these on top of each other, it's hard to make them densely compacted. And that's why unsaturated fats tend to be liquid at room temperature, like this olive oil. Don't get me wrong, olive oil, vegetable oils can have a little bit of saturated fat in them, but nowhere near as much as butter. So you're going to see more unsaturated fats in oils from plants. So yeah, typically liquid at room temperature, like vegetable oils. So what does this have to do with your health? Well, the higher unsaturated fat you have in your diet, the less likely it is you're going to get plaques developing in your arteries, specifically coronary arteries. That's where you end up getting a heart attack. So if you have a diet high in saturated fat, the amount of saturated fat flowing through your bloodstream, in addition to stuff like cholesterol, you're more likely to get fatty streaks, little streaks of fats on the inside of those blood vessels. And over time, plaques can develop that cause the width of that artery to get more and more narrow. And the more narrow, the less blood flow, and inevitably that affects your heart in a negative way. So keeping the amount of saturated fats down in your diet, important. Unsaturated fats, they're a good thing. Lipid catabolism. So catabolism, remember, is breakdown. So lipolysis or lipolysis is the breakdown of lipids for energy use. So when we take, let's say, a triglyceride and break it apart, what happens to the glycerol? Well, the glycerol is converted to pyruvate, which, remember, also has 3 carbons, like glycerol, and it enters the Krebs cycle. Remember, modified pyruvate becomes acetyl-CoA and enters the Krebs cycle. Fatty acids are processed by enzymes to make acetyl-CoA. So you can actually chop up a fatty acid into lots of different acetyl-CoAs, because fatty acids can be like, let's say 40 carbons long sometimes, or less. Beta oxidation, so related to those fatty acids. When you take fatty acids and you break them down into 2 carbon fragments to make this, something amazing happens in terms of the amount of ATP you get out of that. From one 18 carbon fatty acid chain, you can actually get 144 ATP from just that one chain. That's more than from glucose. So you might wonder, well, then why doesn't our body just use fats way more than sugars if it's more efficient? Well, the speed at which breakdown of a sugar happens, way faster. Yes, if you're taking in sugars and lipids, your body will use some of those lipids to break down them into making ATP. But the ease at which sugar is broken down to make ATP is the reason why our body tends to rely on that more. A common misconception about diets is people will just have like hardly any fat intake, but their intake of sugar may be too high. Well, your body will actually sometimes take that excess sugar and it can store it as fat. So you gotta watch your diet, not just looking at no fat, where your calories coming from as a whole. All right, so in terms of making lipids, lipogenesis, the origin of lipids, that's the building of lipid molecules. Some fatty acids, though, can't be built in terms of stringing together that chain and then attaching it to a glycerol, and they're called essential fatty acids, like linoleic acid or linolenic acid. Those you have to get in your diet. You're not making them on your own. The others, yeah. Lipoproteins, the name says it all. It's a lipid protein complex. So when we look at lipoproteins, the first one is actually made in your intestinal epithelium. So as you eat fats, in the lining of your intestine, you're going to make these chylomicrons. And too much of these, yes, your amount of lipid molecules in your bloodstream may be too high. And then inevitably, you end up storing too many fats in your adipose tissue, which is at the deepest part of your integumentary system. Next up, we have these 4. Okay, these 4 all are made in the liver, unlike the chylomicrons. So very low density lipoproteins. If you look at the sequence of these down to LDLs, LDLs and then finally HDLs are going to have slightly more percentage of protein involved. So very low density lipoproteins, intermediate density lipoproteins, I mean, they're important in terms of producing molecules, shuttling molecules. But when we look at LDLs and HDLs, you may have heard about these in terms of good and bad cholesterol. LDLs are known as bad cholesterol, and then HDLs are the good cholesterol. So if you get a blood panel done in terms of testing your cholesterol level, like maybe your cholesterol level is 160, which is actually very healthy, but you may not have a celebration for that if your LDL percentage of that is really high compared to your HDLs. So that's why, in addition to the total number you get for cholesterol levels, they also want to look at what's the HDL number and what's the LDL number. So the reason why low-density lipoproteins are called bad cholesterol is because this is the kind of lipoprotein that takes lipids to your peripheral tissues, like into the bloodstream and to your tissues. So if you're doing too much of that, the cholesterol levels in your bloodstream, bad. But HDLs is the opposite, in the sense that high density lipoproteins, they take fats out of your bloodstream for the sense of storage. And so that's why the bad is bad and the good is good. Depending on the person, this is not the best term to use because somebody could have an HDL level that is actually too high. And just because your LDL is high doesn't mean you're going to have a heart attack soon. So those are just general terms that people look at in terms of measuring HDL versus LDL. So now on to proteins. This is the most abundant organic compound in the body. Like I said earlier in the lesson, if you took all the water out of a body, protein is the next most abundant substance. The building blocks of proteins are amino acids. And like sugars and like lipids, the way that you put together amino acids is a dehydration synthesis. The way that you break them apart is hydrolysis. And it's the same with attaching those 3 fatty acids to the glycerol. So those dehydration synthesis and hydrolysis terms are applicable here as well. They are water soluble as a whole. What I mean by that is your protein as a whole. This is a computer generated model of protein structure. So this thing as a whole is okay with water. The whole thing would be considered water soluble. But there are some amino acids that would be inside of this potentially that are nonpolar. So there are 20 amino acids in nature. Some of them are nonpolar, but the majority okay with water. Many functions. That's why in one of the first slides of the lesson I said that proteins are arguably the most important because they do so much. You can't live without carbs and lipids or nucleic acids, but check out all the things that proteins do. So support, in terms of, on a cellular level, the cytoskeleton that's keeping the inside of the cell stabilized, made of protein. In terms of the connective tissue in your body that's supporting organs as a whole, supporting your body as a whole, it's made of proteins, protein fibers. Movement, so movement within a cell, movement of your body as a whole happens because of protein. Your muscle proteins allow you to move, extremely important. Transport, so not only shuttling molecules through the bloodstream, like attached to a protein, hemoglobin's a classic example, that's a protein, but also transport in and out of cells, membrane proteins, channel proteins. Buffering, if it wasn't for proteins, certain molecules would have a greater ability to harm you in terms of pH levels. So buffering, very important. Enzymatic action, enzymes. There are millions of enzymes. They all are made of proteins. So they are protein molecules that help a reaction along. Hormone synthesis, there are a lot of proteins in your body designed just for making hormones. One of them would be epinephrine, adrenaline, made of protein. and defense. One example of that would be antibodies. So antibodies, which target bacteria, foreign invaders in your body, those are made of protein. Oh, and if you're wondering about this image, that's going to come up in a sec in terms of the different structural levels of protein. You can have these little helical shaped protein parts, and you can have little sheets that are pleated. There's a lot of different shapes you can get from the structural parts of proteins. Amino acids, there are 20 different ones in nature, and that's because there are 20 different R groups. So this is a variable. R, sometimes it'll be an H there. Sometimes it could be something called a benzene ring. Sometimes it could be an OH. There's all of these different R groups here that make the 20 amino acids different from each other. The majority of amino acids can be synthesized in the human body. The others are considered essential amino acids. Just like with lipids earlier, essential amino acids you need to take in with your diet. Peptide bonds hold amino acids together. Once again, it's a dehydration synthesis. An OH on this part here called the amino group, I misspoke. This is the amino group. I saw the A here and thought differently. This is, they call it acid group here, but I've actually seen this more in textbooks. A carboxyl group is the name for this. So CO2H, you can abbreviate it as that. That's the carboxyl group. So an OH on that, and then if you look at the amino group over here, that makes water. So when these two leave, dehydration synthesis. So now let me erase this and show you how the peptide bond forms. It's going to be between the nitrogen of one amino acid and the carbon of the carboxyl group of the next, and vice versa. So this carboxyl group carbon is going to be linked to nitrogen of the neighboring amino acid, and you can get an amino acid chain made of what's called peptide bonds. And amino acid chains are also called polypeptides for that reason. It's just a bunch of peptide bonds stringing together amino acids. So protein structure, there's 4 basic levels of protein structure. The primary structure is, like I just mentioned a second ago, amino acid chains, just a stringing together of amino acids. That's your most basic small little protein. That's what you're seeing here. I know this is in Chinese, but this is a really long amino acid chain. That is a primary structure. And they're zooming into these 4, showing you what amino acids they are: cysteine, serine, leucine, phenylalanine, and then there's whatever other amino acids here. So that's your primary structure. Secondary structure, you saw some of them in that previous image. When you make bonds between the atoms that are on these amino acids and make some kind of shape out of that amino acid chain, like that specific shape might be a helix, that shape might be a beta pleated sheet, like pleats in pants. It's the same concept in terms of like kind of little folds. That's how you get the secondary structures. So you get a lot of different kinds of shaped proteins, different protein functions by having different combinations of helices, beta pleated sheets. Sometimes these are called alpha helices. The point is that bonds between the different parts of these secondary structures is how you get from primary to secondary. Then tertiary structures. If I take a bunch of secondary structures and put them together and have them bonded in a way and interacting in a way where it's now a bigger, more functional protein, that's how you get tertiary, the 3rd level. So it's the interactions from the polypeptides and the surrounding fluid near it, as well as R groups. So the specific amino acid chain within the secondary structure, once you put them together, that's going to impact your tertiary structure, especially from a kind of bond called disulfide bonds or disulfide bridges. So if we had these two attached to one another and there was sulfur helping connect them, that's how you would get this disulfide bond between the secondary structures to make the tertiary structure. And finally, taking tertiary structures and putting them together, you get a full-fledged protein. By full-fledged protein, I mean something like hemoglobin, which is in red blood cells, or something like catalase, which is an enzyme in the liver. So the quaternary structure is how you get your completed protein with a certain shape matching whatever it's supposed to do, whether it's a catalyzing reaction or a structural protein, whatever. All right, we're done with protein. So vitamins. What are vitamins? Well, they're required as vital nutrients in limited amounts. So in your daily intake, carbs, lipids, proteins, trace amounts of vitamins are sometimes all that's required. And some people will take a multivitamin daily. Some doctors will joke that you're just paying for expensive urine. And when it comes to the urinary system lessons, you'll see more about that. But the reason why they say that is these vitamins, and I'm just listing a few of them, if you have a well-balanced diet, you're taking in enough vegetables and whole grains, etc., different protein sources as well, you're going to get these vitamins in reasonable amounts in your body, and you may already be excreting some of them out via urine as waste. So people who have a well-balanced diet and are taking a multivitamin every day, they don't necessarily need it. But if it helps you sleep at night, then do it. So when we look at vitamins in terms of their structure, some of them are fat soluble, meaning not water soluble, and others are polar, meaning they do dissolve easily in water. Vitamin A. Actually helps form epithelium in your body, which is found in virtually every organ. And you've also heard about the eye thing, like carrots is one source of adequate amounts of vitamin A. It's not going to give you your sight back, like I need glasses. My sight's not going to get better. But in terms of keeping your retinal health up there, the retina is very important. Yes, vitamin A is good for that. Vitamin D. Not just from diet, you're not going to get milk as your only source of vitamin D or something like that. You actually, from getting enough sunlight, from enough UV radiation hitting your skin, will make vitamin D. Vitamin D helps you absorb calcium, so it's for bone growth, for maintenance of bone health, as well as joints. So without vitamin D, you're not going to get enough calcium absorbed into your bloodstream. Vitamin E actually prevents the breakdown of vitamin A and certain fatty acids. Vitamin E, not always remembered as much as these two, but important. Vitamin K helps with clotting factors. If you do not have enough vitamin K in your body, you're talking some kind of blood disease potentially forming. So vitamin K, need that. Water soluble, B1, B2, B5, B6, B12, that's most of the B vitamins. You get these mainly from meat and milk. Some of them from bread, like B1, but meat, a common source of these. Not just meat, but let's say you're vegan. If you're vegan, talk to your doctor about some other sources for these vitamins. If you are completely excluding meat and eggs and so on and milk from your diet. Niacin, important. Folate, important. I've actually read that folate in a woman who's pregnant early on, even before she knows she's pregnant, folate's extremely important for the development of the embryo in terms of development of the spinal cord especially. So low levels of folate early on in pregnancy makes it more likely a baby could be born with something like spina bifida. Niacin, without niacin, you wouldn't be able to make NAD. Earlier on in this lesson, we talked about what NAD plus is, NADH. Without niacin, you're not going to be able to make that, and it's a very important molecule in terms of aerobic respiration. And finally, vitamin C. Here's a picture of vitamin C. Vitamin C, you've heard about that. Vitamin C is just an important molecule in terms of keeping many different tissues healthy. It's also an antioxidant, helps lower the risk of cancer over time. Not enough vitamin C will give you scurvy. You've heard about sailors or pirates hundreds of years ago getting scurvy. And so they would start keeping oranges on the ship to prevent that vitamin C deficiency from happening. And then we look at minerals. Minerals are really tiny. They're inorganic ions, not organic, released from electrolytes like NaCl, table salt. You're going to get sodium and chloride out of that. Those are minerals. Functions: osmotic balance, keeping the right amounts of water in different parts of the body, not getting too much on one side or too little on the other, or losing water or gaining water. Minerals are important for that osmotic balance. Tissue maintenance and use. Something as simple as like nerve function. Without sodium and potassium, you're not going to be able to have your neurons functioning properly. Cramps is just the beginning. Enzyme cofactors. Some enzymes aren't going to work properly without minerals. So examples, I mean, you've heard of these: sodium, potassium, chloride, calcium, iron, zinc, copper. If you do take a multivitamin, you're going to find that some of these things are going to be included. Sweat is one way that these come out of the body, not just through urine, but sweating a lot is going to release some of these minerals or electrolytes. And that's why, you know, people will sometimes drink those sports beverages, which also have sugar in them, but sports beverages to regenerate that. You know, people who get like a muscle cramp, they'll say, eat a banana. And the reason why is like, yeah, there's a high amount of potassium in a banana, and a muscle cramp may be due to just overuse of the muscle, maybe due to sweating out some of those particular minerals, and you want to replenish that to get back to normal. So in terms of a balanced diet, I could talk for a long time about this, but I'm just going to highlight some main things. When you look at a food guide pyramid, you're going to see different examples of them depending on what source, but you're going to see grains is making up a lot of what you should be taking in: whole grains, from breads, plant products. Vegetables. Speaking of plant products, vegetables and fruits not only contain a lot of important vitamins and minerals and natural source of sugars, but also fiber, very important, which we're going to discuss in the next slide. Americans in general do not eat enough vegetables and fruits. A lot of people do, but in general, that's something that the average American diet is missing. And studies have shown time and time again that a diet high in fruits and vegetables makes it less likely to develop cancer. You know, keeping your tissues healthy, antioxidants, etc. Dairy, you know, milk products. Some people refuse to drink milk products or eat cheese, yogurt, etc. But dairy does provide certain vitamins and minerals that are important. There are other ways to get that. And then meat and beans. You don't need meat. I know plenty of vegetarians and vegans who are healthy. But meat is an easy way to get iron into your body, to get essential amino acids in your body. But moderation is key. Not too much meat. If the amount of meat you're taking in exceeds grains and vegetables, constipation is one problem you're going to have, but you're probably going to be malnourished in some way. Beans is a great source of protein as well. Not as much protein as jam-packed in meat, but beans, a good source of protein. This is actually not your typical food guide pyramid. This is a vegan food guide pyramid. And if you look carefully, you'll see that there's no animal products involved. So this is a way that someone who is vegan could keep on top of it in terms of making sure they're getting the right amount of nutrients and vitamins and minerals. Oils, you're always going to see at the top in terms of the least amount you should be having per day, because it's just a source of a lot of fat that you're going to get anyways from a lot of these particular products. So some people will make the mistake of having like a giant salad, and salad itself, there's nothing wrong with it, fruits and vegetables, etc. But a salad with a bunch of ranch dressing poured all over it, that's a bad thing with a good thing. Ranch dressing has a lot of oils, saturated fat. I mean, you're getting animal products together to make that ranch dressing, and so that's a source of oils where something like a salad actually might be less healthy than some other option. Using a vinaigrette dressing or you know, a vinegar oil combination dressing is going to be a lot lower in the saturated fats and higher in unsaturated fats. And finally, nutrition facts. People see these almost every day and sometimes don't think twice about it. So I'm going to quickly go through what nutrition facts mean and kind of why they're important. So serving size. All right, if we look up at the top, whatever package you're looking at, and here we've got macaroni and cheese. Serving size, 1 cup. You could mass it to get 228 grams, but just that, you know, that volume measurement, 1 cup, and there are 2 servings per container. So if you consume the entire box of mac and cheese, you've got to multiply all this by 2. Some people forget that. They look at the nutrition facts, like, oh, it's only 3 grams of saturated fat. No, it's actually 6 because you ate the whole thing. I've seen this even on bags of chips, little bags of chips, where people, they're going to eat the whole thing, typically. I've seen little bags of chips where it says 2 servings per bag. And so it's kind of misleading the person. They got to look carefully, and they might have to actually double everything in terms of what's in there. So serving size and servings per container, important. So all of these measurements have to do with consuming 1 serving. Calories. So per serving, this has 250 calories. Calories is a way of measuring the amount of energy contained in it. The calories can come from fat, they can come from sugar, etc. So calories from fat, almost half of the calories are from fat, close to half. So that's important to consider too. There was a law passed in California recently that makes it where all the franchise restaurants have to put the amount of calories on the menu. That's great. I think that's great that people can see that, but what they don't have to tell you is the amount of fat calories. So something may be 100 calories, like, oh, that's not much, but it might be 70 calories from fat inside of there. So it's important to consider what percentage of calories are actually coming from fat, especially if you're trying to lose weight or watch your weight. So when we look at fat, the percent daily value basically means like, hey, if you're on a 2,000 calorie diet, which is kind of average, the amount of calories you're taking in per day, if you want to have the right amount of fat and cholesterol and carbohydrates, by the end of the day, pay attention to the percentages. So if you have 12 grams of fat from this serving, that's only 18% of what you're kind of allowed to have in terms of maintaining a healthy balance. So you still can go another 82% in terms of what's left. You don't typically see the percentage for protein. Protein needs really depend on your activity and musculature in a sense. But when you look at fat, cholesterol, sodium, carbohydrates, they give you a guide here in terms of the percentage. In general, they say, hey, 5% or less, that's low. So if you're looking for a low-fat kind of option and you see 20-something, 30% over here, that's not a low-fat option because 20% or more is high. For instance, when you look at soup, when the sodium levels in soup, most cans of soup I've seen in the store, you're looking at like 50, 60, sometimes even greater percentage of the amount of sodium in the soup. And they're using that sodium to flavor it. But you got to watch your salt intake. So fats, when you look at fats in this yellow section, saturated and trans. So the total fat, 12 grams, total fat. That includes saturated, trans, unsaturated, and then there's different types of unsaturated fats. There's polyunsaturated, monounsaturated. They're not listing those here, but I have seen on different food products where they actually say, hey, here's the amount of monounsaturated or polyunsaturated fats. So saturated fat, it's only 25% of the total, 3 out of 12. Trans fats, very unhealthy. Some restaurants have decided to completely eliminate trans fats. They've been linked to heart disease. A little bit now and then is not a big deal. But if you are, watching your cholesterol levels, heart disease risk, trans fats, not good. So there's 3 grams in this mac and cheese by the time you add all the stuff in. Cholesterol. So cholesterol is lipidy. That is a lipid molecule. Cholesterol levels, it's only 10% of what you're allotted per day. Now, if your cholesterol level is high, like let's say your total number with a blood panel and your LDL is extremely high, doctors might say, hey, you need to cut cholesterol, cut the fried foods, cut ranch dressing, cut all those desserts, those things that are really tasty because they're going to have cholesterol. People like myself actually make cholesterol a little bit more in my liver than other people. And that's a genetic thing from my mom's side. So I need to, watch my cholesterol levels in terms of not going excessive and, eating desserts after every meal. But some people like my brother got a little more fortunate and he can eat a little bit more unhealthy than me and actually have the same cholesterol level because his liver is not cranking out as much as I am. Sodium, specifically, from salt. Sodium, 470 milligrams. about 20%. You're recommended to have around 2,000 milligrams or 2 grams per day. Some doctors even say that's a lot. It depends on the person. If you have high blood pressure and you have physiological problems in terms of your health, you want to limit your sodium, because over time, the more salt you take in, the more likely it is that your blood pressure is going to go up. Carbohydrates, sugar. So hey, when we look at sugars, there's 31 grams. It makes sense. There's going to be carbs in that macaroni in terms of the pasta. There's going to be carbs in the milk, in the cheese, etc. So carbs, 31 grams. That's only about 10% of what you're recommended to have per day, so you can go 310 grams. Atkins diet would be one of those diets where you're not supposed to have carbs. That's not a healthy option for everybody. So I would talk to your doctor first before going on a no-carb diet. That's an option for some people to lose weight easier. But for most people, having some carbs in your body every day is a good thing, but moderation, of course. Dietary fiber, that's a kind of carbohydrate that we don't actually digest and absorb. It comes from typically cellulose. So cellulose is in plant cell walls. So when you eat something like celery, vegetation, it's like Drano for your intestines because you're not absorbing all that stuff. It's kind of sliding through, and it's good in terms of keeping you regular with trips to the bathroom. Number 2, people who don't have enough fiber in their diet are going to get hard stools and more likely to get constipation. So fiber is good in terms of maintaining intestinal health. And then sugars, these are those digestible sugars. You notice that there's 31 grams of carbohydrates. Sugars here might be just simply glucose, but you can see there's 31 total grams of sugars. And then protein, 5 grams. Protein is very important in your diet, of course, because not only, for maintaining muscle mass, but you make so many molecules from proteins in your body, and those amino acids, you don't want to get low on those. You can see, of course, that at the bottom there's vitamins, and depending on what vitamins are in there, they'll list those. Notice there's no vitamin K listed here, no vitamin E listed here, and that's because this dish is not a significant source of those vitamins. And you can see that they adjust here in terms of, like, if your total calorie intake is actually 2,500, well then, since your total calorie intake is more, then you can take in more grams of all these different organic nutrients. If you go 3,000, of course you're going to be able to take in more. But it depends on your level of activity and your health. Somebody who's an Olympic athlete, if they're working out 6 hours a day or more, then maybe a 12,000 calorie diet is appropriate for them in terms of their metabolic needs. But for someone like me who's not an Olympic athlete, I think 2,500 would be good enough. So thank you for watching Educator.com.